Different proteins are made in the cell, different markers are present on the surface. Don't memorize individual ones. I've mentioned the important ones, you know, the surface antibody and so forth. But they change. And there's a special progression during that maturation process. And if you notice, transcription factors, these are all active in the mature cell. A lot of things that were active during the maturation process are not active or present in the mature cell. Okay? Big picture. Oh, and this is in the very end of that chapter. It's like an appendix at the end of that chapter. I'd recommend you look at this a little bit just to work through it. Just get a feel for it. This goes through B cell development. And again, how it develops in relationship to heavy chain and then light chain and so forth. This goes into different surface markers, different CD markers, different adhesion markers. And this is one, this not only goes through development, here we have the immature B cell that's still in the bone marrow. And here we have the mature, naive B cell. That means it has not yet been exposed to antigen, right? When it goes into circulation, it can have IgM and or IgD on the surface. And it goes from one lymphoid tissue to another, just cruising, right? Looking for the antigen, and it's free antigen. Not processed, it's the natural shape of that antigen structure. Okay, this takes it through activation. So this is why I want you to look at this one. This is naive, has not come into contact with the antigen. But here we have a lymphoblast. This has come into contact with antigen. We have alternative splicing. Please look at the endoplasmic reticulum. Those little lines refer to endoplasmic reticulum, rough ER. It's secreting IgM. IgM is the early antibody. It's the first one that's secreted. Remember that graph? IgM is the first one that's secreted. IgG is a recognizable concentration later. Okay, so it's making IgM, a memory B cell. You may have switched to either producing IgG instead of IgM or Ig alpha or Ig epsilon. It may have done the class switching, and it's done that by the time it's a memory B cell. Okay, everyone okay with that? And it has surface antibody. They're just waiting to encounter that antigen another time, so it can go into the rapid response. Here's the plasma cell. Plasma cell, again, it has a lot of rough endoplasmic reticulum, more than the lymphoblast. The lymphoblast is dividing, okay? It's dividing, and then it becomes either a memory cell or most of them become a plasma cell, right? Plasma cell does not divide anymore. Does it have, oh, this is interesting. Does it have surface antibody? Does a plasma cell have antibody on the surface? It looks like it does here, but it's being secreted. It's not shown here attached to the membrane. It's secreting bukus of antibody. But if you notice on this memory cell and on this lymphoblast, it doesn't show it on the lymphoblast, but it has, let me see, I'm going to have to go back to the previous slide. No. Ah! Sorry. I'm not to be trusted with the remote control either. There is no surface antibody on the plasma cell. And I want you to think that through. You have surface antibody on the mature naive B cell. You have surface antibody on the memory cell. I think the lymphoblast may lose it in time because most of the cells, most of the lymphoblasts are making the transition from this activated lymphoblast to a plasma cell. But what's the function of the surface antibody? What does it bind? Antigen. Why, what happens when it binds antigen? It becomes activated, right? You need to have surface antibody present on the mature naive B cell and on that memory cell, because those are the two types of B cells that need to be activated if their specific antigen is present. But on this plasmablast, what's the future for a plasmablast? It's going to die after a couple of weeks. Does it divide? No. It secretes antibodies. Does it need to be activated? No. It's an end of a line cell. So it does not express surface antibody. I want you to know that. I know I really belabored that point, but I really want you to know that a plasma cell does not have surface antibody. It doesn't need it. It doesn't need to be activated. Okay. Memory cell? Yes. How else is it going to recognize that antigen? Okay. Does that make sense? I hope. Okay. We still have the T cell to go through, but we'll save that for next week.